Hello, so today I'm going to be taking you through our NCBE PCR kit. So the first stop is to go through how you collect DNA from your chosen plant samples. So you should all have an FDA card in your kit, like this. And you then obviously need to select which plants you want to try and extract DNA from in order to do your PCR. So I've got spinach, stringless bean, a leaf from a spring onion, and some grass. Now, you need to take a small bit of these plants so that it fits nicely into one of the quadrants on your FDA card. Now you want it to fit nicely in the middle of that, so there's still plenty of room around it, because you don't want it squishing out into the other quadrants, otherwise it will contaminate your other plant samples. So you put it in the middle of one of the quadrants, and then using a pestle from a pestle and mortar, you want to put the cover back down and then, just like I am here, squash the plant sample onto your FDA card. Now this will be transferring all of the chloroplast onto the FDA card. And what you're looking for is a nice green patch like this. And you just peel off any excess. And see, that's transferred really nicely there with the spinach. So I'm now going to try a bit of the stringless bean. So you only need a small bit. And again, pop that down onto another quadrant in the middle there, and then again using the pestle, squash that down. Now the FDA cards that we're using here are excellent for extracting DNA of most plant samples. They've got various components within them, such as SDS, which is a detergent used to break open the plant cells. They also contain EDTA, and this binds metal ions, and the metal ions are actually required by DNA degrading enzymes. So if the EDTA binds these metal ions, then it stops these enzymes from being able to work. The FDA cards also contain uric acid, and this protects the DNA from degradation by free radicals. So I'm now just moving on then to the spring onion. So hang on, I'm not happy with exactly where that is in the quadrant. So you want to be careful basically where you position these. And if you're not happy, then just go back and reposition before you start using the pestle to transfer the DNA onto the FDA card. So that's worked well now for the spring onion. And so then lastly, I'm just going to move on to the grass. So now the grass works really well in this PCR kit, but you do need to be a little bit careful with the grass you select. Now if your piece of grass is a little bit dry, it doesn't transfer very well onto the FDA card. Now unfortunately this piece of grass is a little bit on the dry side. Now that doesn't mean you can't still use it, but it does mean you have to work a little bit harder to get the chloroplast to transfer. And you can very much see when the chloroplasts have transferred, because the FDA card looks green after you've squashed the grass onto it. So I'm not quite happy with how much is transferred onto it, so I'm actually just going to move it slightly onto a slightly different bit on the quadrant, and try again. Now I'm actually still not happy with that, so I'm actually going to use another little bit of grass just over the top, and just try and really maximise on the number of chloroplasts that are going onto the FDA card. Now, once this is done, I obviously need to label which plants I have put into which quadrant, so that if you do store your FDA card, and you can store these in foil bags with a desiccant sachet, and they store really well. But if you do decide to store it, or just when you come to do PCR later, you know which one is which. So I'm just going to write on then my spinach, stringless bean, grass, and the spring onion. So now what you need to do then is fold back the cover, and then this will help stand up your FDA card, and this needs to dry for at least 1 hour at room temperature before you go on to the next step.